to the first in a series of lectures, What's New in the Fall of the Roman Empire? If you like this installment in our new series, we hope you'll come back for the next one to be held on Thursday, December 1st, at 5 p.m. at CGIS on Cambridge Street. More will follow. You'll see it on the website. As always, there will be a reception. I know there's a shortage of food for our undergraduates, and I see some of you here. There will be a reception next door in the Tickner Lounge as soon as this is over. So bear with us, and there'll be reward for your virtue. Tonight, we hear what's new in the fall of the Roman Empire from Professor Kyle Harper. Nature did it. Romans, ecology, and the global history of infectious disease. There is something new in the biggest and oldest of historical questions. In fact, I hope you'll come away from this meeting convinced that there's a lot that's new. All aspects, not just the Roman Empire, but all aspects of the human past are in the process of undergoing revolutionary change, revolutionary expansion in the methods, in the questions, and in the new data that is allowing us to approach and understand the human past in unprecedented ways. These are the goals of the Initiative for the Science of the Human Past at Harper. We wish to bring the power of 21st century science to bear on the greatest questions of human history, and by human history, I certainly mean deep history, the whole of human history, back to our origins, our distant origins in Africa, to bring the power of all of the sciences to bear on the exploration of the human past, and in so doing to create new knowledge, not simply reconfiguring the furniture on a, I would say, sinking ocean liner of knowledge, but on a swiftly progressing ocean liner of knowledge, to create new knowledge of the human past with implications for our understanding of the human present. And in so doing, engage not only scientists and scholars, but students. I see a number of students here. How many undergraduates do we have? Yeah, look at those. I knew they were for knowledge. Thank you for coming, guys. To engage students at all levels in the creation of this new knowledge. To create, through our work, a critical mass of researchers, a network of researchers spread in all the different areas of human investigation and working together in the service of history and the sciences alike. And in so doing, to bridge the celebrated divide between what up until now have been two cultures, the humanities and the sciences. I'm Michael McCormick. I have the privilege of chairing the steering committee of the Initiative for the Science of the Human Past at Harvard, which draws on distinguished humanists and scientists from across the university, from multiple divisions and multiple schools in the one Harvard where we have the privilege to teach and to research. Tonight, we're going to try and bring that power of the science of the human past to one of the big questions of human history. What happened to this Roman Empire? What happened to this extraordinary civilization, this government, this array of institutions, these cultural values that encompass 30-plus modern nations, multiple climate zones across the latitudes and longitudes of greater Mediterranean basin? We're going to ask what happened to the Roman economy through the course of this lecture series. We're going to be talking about the great migrations that played such an important role in history in this era, as indeed they are playing in our own era, as we can see every evening on the television and on the internet. We're going to wonder about the population. Was the Roman population growing? Was it declining? What was its health? What can we know about its health? What can we know about disease in this ancient period? And what kind of implications might that have for our understanding of what people call the fall of the Roman Empire? And finally, the environment, the natural environment of the Roman Empire. Did it change? And if it changed, with what sorts of consequences and implications? These are the questions that we'll be addressing in the course of the lecture series of, I think, 4 lectures at least are foreseen at this point, and we can follow Marcus Aurelius on the way forward to trying to answer those questions. To answer those questions by capitalizing on the scientific revolution and all that it brings to our understanding of the human past, through archaeological science, through the life sciences, through computer science, through the environmental sciences. These are the various disciplines which are making really important discoveries and contributions to a new understanding of one of the most awesome and sinister and terrifying processes in human history. We have no definitive answers, although I think Professor Harper is going to take us quite a step closer, but we have lots of new data. Unbelievable amounts of new data. Only what, a month or 2 months ago, the entire genome of the bacillus, Yersinia pestis, that struck the Roman Empire was reconstructed in robust fashion from victims who died of this, a young woman, a 25-year-old woman who died of this terrible infection in the 540s or 550s in Bavaria, where we never knew that it had struck before. So new data, new ways of thinking, new ways of asking new questions about a very big and very old problem. And we're going to begin in the best of company with Kyle Harper. Professor Harper is a native Oklahoman who got his bachelor's degree, summa cum laude, from his beloved alma mater, the University of Oklahoma, in 2001. He was originally attracted
first things first, is the sound okay? Does that work? Okay. Immediately following that, thank you, Mike, and also I will pay you back for that in some fashion. That is, of course, impossible to live up to, but you're very kind, and thank you all for being here. And let me just begin by expressing my gratitude for being able to be a part of this lecture series. It really is an honor to be able to help inaugurate this year's conversations publicly in the science of the human past. And I am such a believer in what this program represents and what it does. It's so challenging in so many different ways for those of us in the humanities and social sciences to really roll up our sleeves and work side by side with our colleagues in the natural sciences. But it's so important, it's so exciting, and this is where it's happening. And I really feel privileged to have been a part of it, to have been around it for so many years. And it's great to be back in an environment that has nurtured me, a building that brings back so many fond memories, and it's been such an important part of my life. So it's wonderful to be here and to be a part of this. Thank you, Mike, for having me. Let's start with the kind of conversation that cultured, aristocratic inhabitants of the Roman Empire, maybe early in the 2nd century, would have had, that we know thanks to the writings of the great Plutarch, more famous for his lives of the Greeks and Romans, but also the author of extensive tracts on moral conversations. And one of these moral conversations poses among its interlocutors the question, can there be new diseases in the world? And the answers that are given to this question are interesting. One of the participants in this conversation says, no, there can't possibly be new diseases. Nature is complete, perfect, unchanging. The ancient authorities couldn't possibly have missed, like Hippocrates couldn't have missed a new disease in the world. The other interlocutor says, no, there are new diseases, but the reason he says why there can be new diseases is interesting. He says it's because of all the hot baths we're taking in this luxurious Roman Empire. It's because of all the fine wines and foods that we're drinking, and the way that those distort our humors, throw our bodies out of balance, and cause new kinds of diseases. So there can be new diseases in the world. But I want to be clear, even though I think there are new diseases, it's new ways of distorting our body. There are no particular seeds of diseases. There aren't, we might say, germs, spermata of diseases. It's one of the most poignant errors, I think, in all of ancient literature. Poignant in so many ways because within just a generation or two of this conversation in which one of the interlocutors says there can be no such thing as new diseases because diseases don't have their own germs. One of the most important germs in the history of human civilization appears. Oops. What we now call the Antonine Plague. It's a pestilence that erupts around the year 165, in so many ways at the absolute apex of the Roman Empire. It's the very year that the Roman armies have stormed deep into Parthia and destroyed the greatest enemy of Rome, the Parthian state, and one of the 2 or 3 most powerful, along with Han China, political formations in the entire world that the Roman armies make short order of destroying. The Roman army is absolutely unmatched in its power. And the Romans believed that deep inside Parthian territory at a very Hellenistic city of Seleucia, that Roman soldiers sacking a temple of Apollo unleash a vapor, a miasma, that follows the Roman troops back to the Roman Empire. It's a temple of Apollo, and they believed that it comes to cast a plague over the entire Roman Empire. We're extraordinarily fortunate, and I'll come back to this point, that in many ways the greatest medical writer of the ancient world, certainly the most prolific in terms of his extant writings, the physician Galen, is contemporary with this disease event and provides us really extraordinary insights onto a disease whose spatial impact, a point that I'll come back to, is truly out of the ordinary, and a disease whose mortality impact is still debated, even though historians over the last 20 years, it's exactly 20 years since Richard Duncan Jones published a truly seminal article in the Journal of Roman Archaeology, asking historians to pay more attention to this disease. And indeed, in the interim, historians have, and increasingly see it as, if not a turning point, a major point of rupture in the history of Roman imperialism. And regardless of exactly how many Romans we think are killed by this disease event, it is, on almost any reckoning, one of, if not, I would argue, the single largest mortality event in the history of the human species up to that time. And it strikes the Roman Empire, which is a truly extraordinary political entity, at the moment of its zenith. And the Roman Empire stretches across extremely different ecological zones. It stretches from the northern frontiers of Britain to the southern frontiers of Egypt, from Syria to Spain. It integrates these over a course of hundreds of years into a single political organism. The Roman Empire, at the time the Antonine Plague strikes, is home to maybe 70 million people, a quarter of all human beings alive at the time. Its center is the city of Rome itself, which is a city believed to have a population over 1 million, the only Western city to have 1 million inhabitants before London in 1800. It's an empire that has seen robust economic growth. And this is a wage series that I published earlier this year in the Journal of Economic History from a Roman province, from Egypt. This isn't from the political center of the empire. This isn't the extraction of the provinces coming to a privileged center. This is the provinces, and these are ordinary, this is unskilled wage labor. These are literally donkey drivers, dung haulers, fertilizers, the most kind of ordinary unskilled labor. And you see that over the decades of Roman rule that even their wages, their well-being rises because of the extraordinary trade, the extraordinary technology and technical diffusion that the Roman Empire facilitates. Even ordinary people had, I believe, experienced a kind of economic lift by being a part of the Roman Empire. So all of this is progressing down to the great interruption of the Antonine Plague. But the Antonine Plague, if we zoom out and look in terms of the long trajectory of Roman history, is an exceptional event at the time, but it really comes to be characteristic of an entire period of Roman history. We might think of the later Roman Empire or the period of late antiquity as the age of pandemics. This is the first of at least 3 great continental scale disease events. The Antonine Plague, which I'll contend is smallpox, variola major, a virus. The Plague of Cyprian, the most important event in Roman history that no one talks about, in the middle of the 3rd century, that coincides with the most rapid disintegration of the Roman imperial political and economic fabric in the entire history of the empire, whose agent is entirely unknown, but may have been a hemorrhagic fever, and rampages across the Roman Empire for 15 years. And then, of course, the grand climax, the Justinianic Plague, now securely identified as the bacterium Yersinia pestis from ancient DNA evidence sequenced in 2 different sites in Bavaria. What is the first of 3 great global pandemics of Yersinia pestis, a bacterium that is uncannily evolved to be a terrible nemesis for humanity. And so this is an age of pandemic diseases. And this extraordinary sequence of biological events actually leaves not just a small, but actually a significant imprint on the history of human population across the globe. And if we look at the road to the first billion, which human populations reach the first billion around 1800, only 200 years ago, the impact of the Roman pandemics, and then, of course, the second plague pandemic, the Black Death, are really visually striking, just the impact of these mortality events. They're some of the most important events in the history of humanity, and their mortality tolls are evident to see. These are events that historians have paid attention to through the traditional sources that we have at our disposal. Literary sources like Galen, papyri, documents, inscriptions, even archaeological evidence that can give us insight into the patterns of economic growth and development, demographic growth and contraction in Roman history. But what I want to do tonight is to say some of the ways that I think that the revolution of evidence that is represented by the science of the human past, and in particular, the avalanche of genomic data that historians now have at their disposal, if they're willing to enter into conversation with biologists and natural scientists, the ways that those can help fill in some of the dark spots in our image of an event of this extraordinary magnitude. And so I want to talk about genes as historical archives. as well as the way, more conceptually, that we might imagine the history of diseases, and in particular the influence of a model of what's often called emerging infectious diseases. New diseases that, because of ecological circumstances, evolve under particular circumstances at particular moments in history. In short, I argue that we should take smallpox seriously. We should think from smallpox's point of view as an organism that has an enormous influence on the history of the globe. And so historians have been, for at least a generation, doing extraordinary work in global environmental history. And I think this belongs to that body of work, but also is enriched by what's happening in the last 5 or 10 years, which is the advent of extraordinary new evidence in the form of genetics as well as other kinds of evidence about the past of the physical and biological Earth. So you could talk as well about climate as global environmental history, but today I want to talk about genes. And this is the kind of thing that as historians we go to graduate school and spend years training to do. And really, I always think Galen sounds better with a little jazz in the background. So we'll just roll with it. But Galen's Methods of Medicine, here you see a 15th century manuscript in the British Library showing one of the most important, kind of the masterpiece of Galen that tells us important things about the Antonine plague. But on the left is something we might not immediately think of as a historical archive or a piece of historical evidence. It's a smallpox genome, one of, I think, 46 that are easily available on the NIH's website, and that preserves extraordinary information that's interesting to those of us who are concerned about what happens to the Roman Empire. Microbial genomes are archives. They tell us historical information. They tell us both functional information about what this organism, which is a virus, does, the kinds of proteins that it can build, the kinds of particular weapons, virulence factors that it has to attack immune systems and tissues in the human body. But it also preserves information about the way that this particular virus has impacted history. It tells us about its evolutionary relationships, its phylogeny, its relationship to other similar related organisms. It tells us something about its age. If we can know something about the rates at which this kind of virus mutates, we can know something within certain boundaries of error about when it evolved and how it evolved. And that information, in turn, can tell us something about where it evolved. And all of this information will be critical in helping to fill in our picture of the Antonine plague. And I think, even more broadly, all of this is contributing to a conceptual revolution in the way we think about viruses, bacteria, other microbial parasites. The way that the ecology, including the ecologies that human societies have built, impact their evolution, and the autonomy, the role, the agency of these organisms in human history. I'll come back to that as well, but the microbes have a huge role in human history, and they are all around us, and the genomic revolution is helping us to see that. And they help us approach what is, I think, one of the most basic questions, one of the hardest questions that can be asked, which is what killed the Romans? Mortality patterns are one of the most powerful forces in human destiny. And I remember a long time ago, I was just out of graduate school, I was at a conference and I asked a very good medical historian, this was over beers, after the conference was over, I said, if I twisted your arm and made you guess what was the #1 killer in the Roman Empire? And I remember, the Romans, this great civilization, you think of the Colosseum, this mighty empire, and I was crestfallen when he said the #1 killer of Romans was probably just little diarrheas. It seems incommensurate that the worst enemy of this mighty civilization was something like Salmonella enterica that you see here. And you see very deliberately a Roman latrine from Ostia, a public latrine from Ostia. But these are the worst enemies of the Roman Empire in a world where infectious disease is the #1 cause of death. Not violence, not chronic diseases like most of us in the modern world die from, cancers and cardiovascular disease, but infectious diseases are the #1 cause of death in the Roman Empire. And so it's little things like Salmonella that have the strongest influence on the Roman health and life outcomes. Historians have worked for decades on life expectancies in the Roman Empire, and somewhat disappointingly, we still don't really know, although most of the most disciplined guesses place Roman life expectancy in the early to mid-20s. It's, in our terms, an underdeveloped society. It's a high mortality society. What we do know is that health outcomes in the Roman world are quite bleak. And one measure of health outcomes is human stature. Stature outcomes are determined both by nutrition and by disease burden. Metabolic costs of running an immune system in a high disease burden environment is extremely high and is known in a number of historical societies significantly to depress height. Obviously, nutrition is the primary influence, but in many historical cases, the disease burden is a huge influence on health outcomes. And there is no doubt in my mind that the Romans, for all their greatness, we can say with confidence, are very short. They're extremely short people. Here's a meta-study I did. We could go into, I think there are problems in the underlying data, but show you over time that the Romans are shorter than their early Republican or Bronze Age or early Iron Age predecessors. Here's, I think, actually the best study of femur length that's done by one scientist. There's no inter-observer error. And the conclusion is indisputable in this evidence, which has good statistical foundations, that Romans are short. Their femurs are shorter, Romans are shorter than either the people that come before them, the Iron Age people would have looked down on the Romans, or the people that come after them. In the Dark Ages from the 6th, 7th, 8th century, people are tall again. This could be related to nutrition, but the best evidence suggests that, in fact, it's because of the extraordinary disease burden that's created by the disease ecology of the Roman Empire. Another insight into this, and I emphasize that the Romans are the first civilization, I think, where we can get different kinds of documentary insights onto the way that people actually died. And among the most important of these data sets is a huge number of inscriptions that tell us when the Romans died in the year, seasonal mortality data. And in the modern world, where infectious disease has declined significantly as a cause of death, seasonal mortality patterns have leveled out. There are still some age differences in seasonal mortality. The elderly tend to die a little bit more often in the winter, college students in spring break, of course. But for the most part, if you look at seasonal patterns and death in the modern world, it's flat. But not so in ecologies where infectious disease is dominant. And it is striking that the amplitude of seasonal variability in death patterns in the Roman Empire is among the highest known of any pre-modern society. And it's, of course, the late summer and early autumn that are absolutely deadly in the city of Rome. When gastroenteric diseases ramp up in July and August, and then, of course, when malaria in August, September, and even into October, as we know from later records, take their toll on these societies. So the Romans are bearing a huge burden of infectious disease. And there are a number of reasons why I don't think nutrition is what explains the poor stature outcomes of the Romans. But one is that disease is clearly no respecter of persons. It doesn't care if you're an aristocrat who's well-fed, who's well-nourished, or you're a commoner living in the streets. And if you just look at the black line in this, the seasonal mortality patterns of people who are buried in sarcophagi, these are the 1%ers, absolutely the same. The summer and early fall are deadly for them. And I put a coin up of Marcus Aurelius's wife. We've seen his picture a couple of times already. He's a star tonight. This is his wife. She's the real star. She bore him 14 children, only 2 of whom survived their parents. Even for the emperors, the disease environment was truly a tragically human situation. And this coin celebrates the fecunditas of the Augusta. Why is the Roman disease environment so deadly? Well, one is because of the sheer density. 1 million people living in a city has deleterious effects on the disease environment. People living close together, of course, can sustain other kinds of microbes that can't survive without that kind of density. But the sheer environmental challenge of managing a population in a pre-modern environment of that size is overwhelming. And here you see amoebic dysentery, also probably one of the greatest killers in the Roman Empire. It's estimated that at its height, the city of Rome produces 100,000 pounds of human excrement per day. There's simply no way, even with the marvelous civil engineering of Rome, that they're capable of removing that kind of waste efficiently from the city. And that says nothing about animal waste. It says nothing about the fact that the major implement of hygiene in the Roman Empire is a sponge on a stick that is reused for washing oneself. In the words of a classicist, the hygienic implications of using such an instrument are at best dubious, to say the least. So the sheer environmental consequences of living in such a dense environment in a pre-modern world must have been truly overwhelming. It's also an artifact of the connectivity of the Roman Empire. The Roman economy grows because of the extraordinary levels of trade, migration, interconnectivity, integration, in an empire that, unlike any before or after, unites the Mediterranean into a common trade zone with a single currency, a single set of institutions. It's truly a historically exceptional level of integration for a pre-modern economy, as Peter Timman will not object to. And the disease consequences of that are as much a part as the economic growth that comes with it. It's also a dangerous disease environment because of what the Romans do to the physical world around them, to nature. Here you see a beautiful reconstruction of what the coming of the Roman Empire means. This is from outside Narbonne. It's a publication that shows the original kind of landscape filled with a diverse hardwood forests. And then as time goes on, as the Romans extract resources and turn them into production, the forests are thinned out. And this is the story all across the Roman Empire. Deforestation, transformation of natural landscapes, which is, if you're interested in human disease, which is an absolute roll of the dice. It coaxes the evolution of microbes. It brings humans into contact with unfamiliar microbes. And it's today one of the biggest causes of emerging infectious diseases, as we'll come to. Everywhere the Roman Empire goes resource extraction and landscape transformation on a massive scale. And they know this. They're, of course, proud of their civil engineering. Pliny the Elder says if you were to make a careful calculation of the abundance of water for the public in baths, in pools, in canals, in urban houses, in gardens, in suburban villas, and along the distance traveled, and the arches built, the mountains sliced, the valleys leveled, he would admit that nothing more marvelous exists in the world. So the Romans transform natural landscapes, and, of course, the natural climate varies around them as well, all of which stirs the stew of disease evolution. And so the Roman world bears a high burden of endemic diseases, especially local gastroenteric diseases that are transmitted by the fecal-oral route, and as well as epidemic diseases, particularly like malaria, which varies significantly from year to year and is one of the major killers in Italy and in the city of Rome itself, as Galen, the great doctor, is a very important witness. And the disease ecology of the Roman Empire contributes to the spread of new diseases. And this is increasingly showing up in the genetic history of pathogens like leprosy, like tuberculosis, which spread all across the Roman Empire to an extent that they had never known before. But leprosy and tuberculosis are both something interesting. They're chronic infections. They move slowly. They diffuse across the Roman Empire. What you don't see in Roman history, as well documented as it is, down to AD 165, are pandemic events. Widespread biological mortality events with acute, deadly diseases. They're rare. And the reason lies in the constraints of the microorganisms themselves. It's hard to jump from one victim to the next. It's the mechanics of transmission that are based on mosquitoes or based on human waste. And it's the limits once pathogens are in the body. Many of them are evolved to be low mortality, like the cold caused by rhinoviruses, or to be chronic infections like tuberculosis. And there are deep evolutionary reasons for this. Highly transmissible, highly lethal diseases struggle to evolve until humans started creating environments like the Roman Empire. And we can now see more of the entire history, starting to see more of the entire history of microbial life, thanks to technological revolutions. Here from the 1670s is the first microscope that allowed humans to see tiny little microscopic organisms. Here today is what about $250,000 will buy you a high throughput gene sequencer that lets us see the entirety of the microbial, diverse microbial world that exists around us. And truly, it's their world. We're just happening to live in it. They've been here for 3.5 billion years. There are maybe upwards of 1 trillion microbial species. There are 40 trillion bacterial cells alone on or in the human body. Most of them are totally harmless. They're living in the environment freely, or they're harmless, they're commensal. They just happen to live on or around us, or they live in animals and are incapable of really doing us any harm. So if you think of the trillion species of microbes that may exist, most of them don't have the biological tools, virulence factors, that let them invade, stick to, or beat our immune systems. There are only around 1,400 known human pathogens, very few of them viruses, around 500 bacteria. That's actually a tiny number, and most of them are relatively self-limiting. They aren't even capable of causing biological events on a multi-continental scale. However, we live in a world where we're increasingly sensitized to those possibilities. The world of emerging infectious diseases that exists around us. The rise of new or newly emerging diseases like Zika, Ebola, and so on, that sometimes evolve in response to the antiseptic environments and antibiotic medical regimes that we create, that evolve amidst climate change that's happening around us, and that in particular, and this is the major source of emerging infectious disease today. It's not antibiotic resistance, it's not climate change. Those are important ingredients, but it's ecological transformation. It's deforestation, it's agricultural exploitation, it's extraction of resources. The most important source of new human pathogens are wild animals. Primates, other mammals, birds that are host to diseases that either are capable of or evolve the capacity to infect humans. And here you see a study of the newly discovered, at least, infectious diseases over the last 50 years, and the importance of zoonotic reservoirs, and in particular, wild animals as the source of these infections. And here you see there are places, of course, on the globe that are more biologically diverse than others. The tropics, of course, tend to be diverse microbially, but also in hosting the kinds of species that are potential reservoirs for human infectious diseases. And so what we need to do is to think about a deep history of infectious diseases, because it's not always the case that even when one of these rare microbes like smallpox exists, that it's really capable of doing significant damage. The basic parameters of the way that human beings have lived for most of their history couldn't even allow the existence or persistence of acute and highly lethal virulent infections like we see today. It takes building a world that's capable of inviting a microbe like the smallpox virus into it, the world that the Romans create. Smallpox is one of the worst killers in all of human history. And it's thanks to Galen's acute clinical notes that we're able to identify the likelihood of the agent of the smallpox, of the Antonine plague as smallpox. He describes in detail the black extrusive pustular rash, the way that the disease progresses across a case history, the importance of different symptoms that are very close to conclusively identifying the disease as smallpox. And in fact, a huge consensus has formed among Roman historians that the Antonine plague is smallpox. It also fits with what we know of the population level dynamics of smallpox and of the Antonine plague. This map in itself is one of the most extraordinary facts about the Antonine plague. There simply aren't many organisms that are capable of doing spatially what the Antonine plague does. It strikes across 3 continents in the space of 2 or 3 years. It's a disease that's directly transmissible between humans. It's spread by virions in droplets that are breathed in. It's extremely pathogenic. It only takes a few virions inhaled to become infected with smallpox. It has a long incubation period when its victims are not immobilized, when they can travel before they know that they even have the disease. It leaves those who survive it immune from subsequent infections, but it has extremely high case fatality rates, probably 40–50% in the context of pre-modern medicine. But smallpox isn't magic. It, like any organism, has its limits, and its most important limit is that while Peter would probably die if I had smallpox right now, if I step a few feet away, he's probably okay. Transmission of microbes is always one of the most important factors in its epidemiology, and smallpox has to be transmitted from human to human in fairly close contact. But it's the advent of whole genome sequencing that's starting to fill in the picture of what we know about this extraordinary killer. Smallpox belongs to the orthopox viruses, and its closest relatives are, here you see the top 6 of these are smallpox genomes. Its closest relative, that TATV, this is a phylogenetic tree. Its closest relative is the tetrodotox virus. It's an orthopox virus that infects only the naked-soled gerbil, which lives only in a particular part of the world, and its next closest relative is camelpox virus. It also tells us something about when smallpox emerges. Smallpox isn't a disease that has existed time out of mind. It's new. It's maybe as young as 2,000, as old as 4,000 years old. It evolved something like 3,000 years ago, give or take 1,000 years. Smallpox is a novel menace in the globe. And here's this cute little thing. This is a naked-soled gerbil, Gerbiliscus kempi, and it is the host of smallpox's closest relative, the Pteropox virus. And the importance of this is that the naked-soled gerbil only lives in the dry forests of Africa. And this tells us something very important about smallpox. Smallpox evolves in Africa 2,000 to 4,000 years ago. We also have confirming evidence for this in the fact that within the contemporary smallpox genomes, the greatest diversity within samples comes from Africa. There's very compelling reasons to believe the smallpox emerges, it evolves in Africa a few thousand years ago. I've left out one critical fact about the way the Romans built a disease ecology. In addition to building a dense, interconnected, ecologically transforming empire, they also connected that empire to the globe in ways that no society before them had. Under the Roman Empire, Indian Ocean exchange doesn't just grow incrementally, it absolutely explodes. And there's been extraordinary evidence over the last 20 years to accumulate showing how profoundly connected the Romans were to the world of East Africa and the Indian Ocean. The excavations along the Red Sea coast have shown the growth of huge trading entrepôts that were critical nodes in Roman trade. This littlest circle there circles what are called the Pharisson Islands, where just 10 years ago, a Latin inscription was found showing a detachment of Roman legionaries that occupied the Pharisson Islands off the coast of the border between Yemen and Saudi Arabia, the southernmost outpost known of the Roman Empire. Why are they there? To protect the trading ships that are sailing up and down the Red Sea. Just about 10 years ago, too, on Socotra Island, were found inside a cave over 200 graffiti of sailors, shippers, traders from all over the Indian Ocean world that show us right at this moment the extraordinary global connectivity that the combination of Roman capital, trade, and Indian Ocean commodities had created. And Muziris, one of the most important shipping centers on the spice coast of India, is evidenced in a papyrus that showed up only about 30 years ago that records the details of a transaction that it's like the equivalent of nearly several $1 million worth of pepper being traded in a single ship in one shipment. Everything we've learned confirms that the combination of Roman capital and wealth, consumer desires for ivory, spices, slaves, gold, and above all, pepper, and increasingly silk, create an interconnected Indian Ocean world. And it's across this world that a virus like smallpox comes in to the Roman Empire. So I propose that we think of the Antonine plague as an emerging infectious disease, a new disease, smallpox, that evolves from a zoonotic reservoir in Africa that somehow hops onto the trading belts of the Indian Ocean world and comes into the Roman Empire. This is a truly global moment. This is a moment when the Roman disease ecology connects with the biogeography and evolutionary history of a particular organism to create one of the most important biological events in the history of the human species. And as a final thought, I think that this way of telling the story that relies on the genetic evidence points us towards a way of rethinking the entire history of human societies and their role in creating evolutionary contexts for infectious diseases. This points us beyond a paradigm that's been dominant since one of the greatest books ever written, William McNeill's Plagues and Peoples, popularized in Jared Diamond's Guns, Germs, and Steel, that places all of the emphasis on the rise of cities, dense environments, where domesticated animals live in close proximity to humans and give us their disease. We've blamed way too much on the cows. The cows are mostly innocent in the story of human infectious disease. And as a correlate of this story, of course, is that societies that weren't exposed to domesticated animals for thousands of years were immunologically naive and exposed to the crowd diseases of Europe and East Asia. So that story, I think, is being replaced by one that places emphasis not on what McNeill calls the civilized disease pools, crowd diseases, but one that focuses on humanity's interaction with the natural world, with the wild, that is the most important source of new emerging infections, zoonotic reservoirs. It's a story that would place less emphasis on cities as disease environments and more emphasis on the connectivity of humans around the globe and humans connected to different ecological zones around the globe. I think one of the great open questions still is the role of natural selection on the human genome in recent times, and the question that is still entirely unanswered of whether different human populations have, in a consequential way, developed different genetic profiles that contribute to differential levels of fitness in the conditions of different disease ecologies that have had consequences on human history. Do different human populations have different genes for different kinds of innate immunity? There's evidence that they do. There's evidence for selective pressures in recent times on those genomes, but it's still mostly unconnected to human history. And above all, I think it is a story of microbial ecology in which we take the perspective of the microorganisms that share the planet with us and for whom we create the evolutionary environment. But above all, we know that it has to be a molecular history, with historians working hand in hand with geneticists, biologists, and other natural scientists to help us understand the story, the science of the human past. And I think it points us towards seeing the Romans not as the end of an ancient world, but maybe as the beginning of a modern world where our ecologies, where the connectivity and environmental transformation that we have created set the background for the evolution and establishment of entirely not just new diseases, but new kinds of diseases, uniquely powerful and deadly germs. And I think it lets us look at old Plutarch not as a hopelessly deluded ancient figure, but as someone who was starting to ask the right question. Can there be new diseases in the world? We're fortunate that we have the tools to say emphatically, yes. Thank you. Yeah, of course. Please.
An amazing presentation that has taught me an enormous amount in very short order. That conciseness and clarity opens the field and the time, the space to pose questions to this remarkable young scholar. The floor is open. Professor Harper has kindly agreed to engage with the questions and challenges you might have for him.
On your latter point, just look at blood types, A, O,
Yeah.
etc., driven by malaria and other pressures,
Yeah.
and has significant different consequences depending on what blood type you have to this day in terms of certain diseases. So.
Malaria is great. I'm glad you brought this up. Malaria is the single best example where there has been selection for genetic traits that confer some degree of resistance to important pathogens. In this case, the sickle cell trait that gives some immunity to malaria. That's the best example. Now, the problem has been that that's sort of been standalone. You can see the whole gene. You can actually see the phenotype. We know mechanically some things about how it makes a difference. And that's sort of been lonely. Hanging out there is the one really good example of differences between human populations that are genetically based that have important fitness consequences in particular environments. It's been really lonely. I think what will increasingly happen, and there's been a lot of good work in the last 5 or 10 years with whole genome sequencing where people are looking for particular genes in the innate immune system that show statistical traits of signatures of recent selection. Those are starting to pop up, and I think but it's still not kind of connected to historical pathogens and human history. That is still, I think, to happen.
Nick Patterson.
I'm Nick Patterson. I'm a geneticist. I'd like to ask a question and a comment about bubonic plague. So I'll give my comment first. Just echoing what you said, and it's a major surprise, people have looked really hard for selective signals of resistance to bubonic plague, which after all, a mass killer. I mean, 30–50% of the population of Europe died, and there were multiple episodes as well. So that would seem like an obvious case where selection that's a comment. The second one is a question, and I'm sure you know, we now have evidence of white pastors on the steppes. You know, not an urban, it's a nomadic, not an urban culture. What's going on, and do you think there may have been major demographic consequences?
So first to your comment, I appreciate the point you make very much, and I think what's happened among historians is that because the sickle cell trait has been so lonely, people have looked for others, and it's been a pretty meager harvest to really find the kinds of genes that you're talking about, and that's why I think this question is still open. And I appreciate your comment. Just your question about Yersinia pestis. So just so people in the room know, a little over a year ago, I think there was a paper by Rasmussen et al. That showed from archaeological number, like a dozen or so archaeological specimens of the Bronze Age, so from 3000 BC, the most recent sample centered on 951 BC, that sequenced Yersinia pestis genomes. And so Yersinia pestis is there throughout Central Asia from at least the Bronze Age. And this is a problem. I mean, it's not neat, but that's okay. That's what keeps us employed. And so I think there's a lot of work to be done still to understand what is the epidemiology of Yersinia pestis in that time period. So Yersinia pestis is different. It's a bacterium. It's a huge bacterium. It's massive. It's got all of these genetic tools. It's like the Death Star. It has all of these little weapons. It's enzootic. Smallpox is interesting. Smallpox is a virus. It's an obligate human parasite. It's why we eradicated smallpox. It has nowhere to hide. We will never, we'll never say never. We will probably never get rid of Yersinia pestis. It lives in rodents. Sometimes we get caught in the middle with terrible consequences. But it was there as Yersinia pestis in rodent colonies, killing humans for at least 5,000 years. And it's very hard to say how that worked. I'll just make two quick points. One is that it's only in the latest of those samples from 951 that Yersinia pestis has the genetic tools to make a protein called Yersinia murine toxin that lets it live in the midgut of fleas. So bubonic plague has to be spread by the flea bite. Those Bronze Age samples, except for the last one, aren't causing bubonic plague in anyone. They're causing pneumonic plague because they can't be spread by flea bite. How humans get them, I don't know. We were talking earlier today, maybe they're eating rodents. There was just a paper yesterday about rodent consumption in the Bronze Age, and then they're spreading it pneumonically, which it doesn't spread as efficiently from human to human.
A Russian archaeology friend of mine says that there are steppe populations today that eat these rodents as
There are, yes, there are. Don't ever eat rodents in Kazakhstan. It's a terrible, terrible idea for many reasons. But I'll just say this one last point about the genetics. We need to look really closely at that latest Rasmussen sample. I think one of the plasmids doesn't code for the form of the plasminogen activator that lets Yersinia pestis drive right through human tissues. I actually think that even the latest of those Bronze Age samples maybe isn't quite yet the monster that the first pandemic is. But what we really want to know is why all of a sudden in 541 does bubonic plague for the first time become this pandemic nuclear bomb? What combination of genetic and ecological factors conspire to let that happen? It had never happened, even though Yersinia is around in pretty close form for thousands of years before it.
I know you mentioned that the seasonal effects and also just the sheer population densities in cities created pretty much a breeding zone for these kind of diseases. But did the immense cultural diversity within the empire cause any kind of niches for smallpox that can cause any that led to any kind of evident disparity among the effects of the disease across the empire? Like, were there any hotspots because of that climate?
Yeah, that's a great question. We would love to have the kind of data that would let us really give a good answer to that question. We don't, and so I think what we have to do is we have to be smart about the way we frame the question. And you make a good point that I would probably like to emphasize if I had more time, which is that I kind of focused on the disease ecology of the capital, but that's very different from the disease ecology 5 degrees north, 5 degrees south. Diseases are really embedded in the local environment. And
Those are unique from place to place. They change over time. One of the things that happens is, the Romans bring germs with them. And so you can actually see in Britain, there's pretty good evidence. The other really good stature study that I trust is from a sample in Dorset in southern England. And they're tall, and then the Romans come, and they're short. And then the Roman Empire falls, and they get tall again. The Romans bring some germs with them. And so the disease ecology is local, disease ecologies change. And smallpox would have flourished in environments that are dense. So it would have been worse in places like Egypt, where because of the ecology of the Nile, people are really crowded in together. It would have been worse in cities like Rome. It wouldn't have been as bad in villages. It certainly wouldn't have been as bad in the western countryside, where the people are settled in dispersed farmsteads, unlike the East, where they tend to inhabit condensed villages. So we just don't know enough to really say exactly how smallpox hits in particular places. We have clues, and I think we can say that it's worse in places where people are living. It's bad in the army. We know it devastates the army because you've got troops living in barracks close together. It's horrible for smallpox transmission. But it wouldn't have been as bad in places where they're all spread out. And going back to the plague, this is what makes Yersinia pestis so devious, is that it's not density dependent. It's not spread from human to human. Principally, it's spread by rats and the fleas that live on rats. And so Yersinia can get deep into the countryside, even where humans aren't living in really dense agglomerated settlements. So there would be differences across the empire, and there'd be differences based on what kind of disease it is and how it spreads.
I'm a microbiologist studying the gut microbiome in part, so I think I'll never have the opportunity to ask this question again. What do we know about the sponge on a stick? Like, how do we know about that?
Yeah, there's some sources about it. The classicist that I cite on its dubious hygienic implications is SCOBY. It's a great article from 1986. You can, it's easy to find online in black market copies because it's the best article written on the disgusting environment of Rome, and it has all of the limited sources that we have for the sponge sticks. SCOBIE. And we're talking about this today with somebody. It's article's 40 years old now. It needs to be kind of updated. But it's mostly from like comic sources that are making bad poop jokes. They're funny 2,000 years ago. They're funny now. We don't know nearly as much as we would maybe like to.
I'm Peter Kemen, economic historian. This connects also to another literature. There are people who've been studying skeletons and have found that they interpret the low height as low nutrition, not as no there. So they've been perplexed by the fact that the Roman Empire seems to be prospering, but the skeletons that we have seem to be short. And you have an explanation for that. But like you're disputing McNeil, you may be disputing also this presumption of the economic historians who are looking at skeletons and giving an entirely different interpretation than you are.
And I'll give you a couple of examples. So I don't think it's unique to the Roman Empire that you have a population, what I call them is rich but sick. So the Roman incomes are high, but their biological well-being is low for ecological reasons, not related directly to economic productivity. That's also the case of the 17th century Dutch, whose wages are higher than almost anybody else. They're tiny. Go look at the Dutch master paintings. They're all like this tall, the people, and that's evidenced in the skeletal remains. And then, as you know better than I, in the context of the U.S., it's called the antebellum paradox. A lot of good debate about what's happening to real incomes in the early 19th century. I think whatever you think about wages, people in the late 18th century in the U.S., some of the tallest people, healthiest people, some of the richest people that have ever lived, and then even Americans get shorter. But so do the English, so do the French. Everybody kind of gets shorter from between like 1800 and 1850 or 1875. And regardless of what you think the wages are doing, I think there's good evidence that the disease environment is the main contributor to that. And so there's some really good papers on the US that show in sophisticated ways that things like being connected to a train line, when you hold a lot of other variables equal, make you shorter because you're connected to the germ environment. And so people in 1850 get short because they're moving to cities. I think their wages, maybe stagnant, but they're getting shorter because they're around more germs. And particularly in childhood, during the developmental period, they're exposed to really high pathogen loads. So it's not just the Romans. I think you have a handful of historical societies where people are getting wealthier, but they're getting shorter. And we know that the biological cost of running an immune system is really high. And if you're sick, if you're just bombarded with pathogens when you're 1, when you're 2, when you're 3, the imprint of that is really profound.
Yeah, actually, I have a question that's kind of playing off that point. So I was wondering a bit about the composition and the provenance of the samples you've kind of been as pre-Roman, Bronze and Iron Age, Roman and post-Roman. Do you think you have substantial, significant enough control over ancestry compounds or class differentiation? Because as Nick knows, and as we've seen from genome-wide data from various contexts, including from the Bronze Age steppe, we have independent signatures of selection for decreased height in Neolithic farmers of Europe and increased height in pastoralists of the Ponto-Caspian steppe. And that systematic ancestry differences, which are still evident today, actually account for phenotypic disparities in height that you can still observe between southern and northern Europe. So do you have adequate control over
No. The short answer is no. And I think we think we should be cautious to put too much independent weight on the stature data. But because there are lots of things we can't control for in the data, above all at present. But I think even in the most optimistic scenario, we can't control for the selection of the population that we're looking at. You just won't be able to control who gets buried and who gets looked at. And so in addition to what you're saying, I think we don't know enough about the entire sample, the parameters of the whole sample, like who is getting in these cemeteries to put huge amount of weight on that. But I think there are more things that could be done, like looking at the genetic composition of the sample. These, I wouldn't put too much weight on these, but they're worth something though, because this, where did we go? This is a really robust look at 20 different sites across the Roman Empire, across Roman Italy, just in Italy. And it happens everywhere in Italy. So you'd have to ask, right, we're limited in the kinds of data we'll have, but you have to ask, okay, well, what would be the best explanation? Could we imagine that there's changes in the population? We could, but there's not considered especially likely in a lot of the sites that are being sampled here. And when you start looking at a huge number of different sites and you see the same patterns over and over, you have to explain it somehow. And so it's just a matter of, in our world, what do you think is going to be the best explanation?
Well, thank you for the talk. So it's sort of a commonplace in the sources, even that Romans are short, barbarians are tall. And if you're right that this is about the different disease pools, do we have any evidence of these barbarian groups when they enter the empire and are exposed to this new gene pool? Do you get the Goths coming to the empire and suddenly they're all dying? Or because that could be a sort of natural experiment.
Yeah, certainly in the context of military invasions, you have, I think, basically credible reports that invading groups are deterred by the disease environment. And so just to give you two specific examples, if you look at the Hunnic invasion, where they get all the way to Constantinople, and then they end up turning around to the 440s for reasons that are obscure. Well, the one contemporary source we have, Isaac of Antioch, says that they all got sick and started throwing up, and they left. That's reasonable enough. The other example is about 8 years later, the Huns again are in Italy in 441, 42, and the pious legend has it that Pope Leo goes out and kind of talks Attila into turning around, but other contemporary evidence says that there was an epidemic. And for reasons that would otherwise, I think, be mysterious, the Huns just leave. They leave Italy, having sacked a few towns in northern Italy. And I think it's entirely believable that you have a population that's basically lived in ecological zones where there's no malaria, for instance. And they come into Italy, they've got horses, they're feeding them right there in the middle of the pastures. The mosquitoes are biting them, they get malaria. Attila says we probably ought to get out of here. So, the evidence is what it is. It's this thin, and you have to wonder, but you hear that enough. What we don't know, what would be more interesting, is when you have cultural groups moving en masse, not just invading armies. Because invading armies, of course, it's not just the lack of prior exposure to pathogens like malaria, where prior exposure is really important. And all the Romans are kind of malaria survivors. I really believe that. But moving armies have different unsanitary conditions. They get typhus. They get all kinds of things going on in the military context. But what we don't know that much about is what happens like when the Goths, when 100,000 of them cross the Danube and move into the Roman Empire. Our sources just aren't good enough.
So how did the epidemics differ, or did they differ at all between the Roman cities and the countryside?
We would love to know. We don't have sources that provide quite enough fine-grained resolution to say everything we would like. But we can say, I think, some intelligent things. When we look at the epidemiology of different diseases, so it's one of the most important reasons in knowing what the pathogenic agent of epidemic is that it tells you about the biology and the epidemiology. Smallpox can't do certain things. Yersinia pestis can't do certain things. It can do other things. And so you combine the knowledge of the biology and the epidemiology of the disease with fragments of historical evidence, just fragments. And it's the synthesizing of those two that helps us get at otherwise really intractable problems, like how many people died. Well, those are estimates, and they're based on a sequence of assumptions, both about what the agent is and the way that Roman society is configured, and the little fragments of evidence we have for what happens in different parts of Roman society. And so the evidence for demographic catastrophe in Egypt is pretty good because we happen to have papyri, and we have few hints that there's serious demographic consequences in Egypt. But Egypt is an unusual environment. You can't just extrapolate, well, what happens in a village in Egypt is what happens in a village in Spain. Those are very different human environments. And so there's a lot of uncertainty. You can also look at things like the second order consequences. And so one thing that I've tried to show is that there are movements in factor prices, that the price of labor, the price of land change in the immediate aftermath of these disease events. Well, that could have other causes. That's not the only possible explanation for those. But if that's what happens, and if we have other reasons to believe that these are severe, then you can say there's a good case to be made that there is a demographic consequence of these biological events, and here's how that might have happened. But there's a lot of blind spots. It's why, for instance, I think that the genetic evidence from Bavaria for the Yersinia pestis, for the Justinianic plague, is so huge, because this is somewhere that was a real dark spot on the map. We didn't know if plague had got there. We didn't know if it had penetrated into these kinds of villages. These are 200-300 people living in these little villages on what is beyond the frontiers of the Roman Empire, and they were killed in the Justinianic plague. If it was there, it was everywhere. I mean, the fact that it's in that kind of place is just astonishing. The fact where we found it and where they sequenced it changes the level of certainty that we can have that the Justinianic plague penetrated far into the countryside, in the West even.
Perhaps the last question, Professor Orlando Patterson.
This is wonderful. I'm sitting here waiting to hear you utter the one thing we have in common, which is slavery, and how slavery is basically others moving out into other places. But of course, the Romans are also bringing in vast numbers of people into Rome itself. And I just wondered whether you've thought about how this is connected with the microbes. Because, just the Romans moving, so taking the disease to them elsewhere, but all those vast numbers of people which actually very seriously transformed the Roman population.
Yeah.
I'm just wondering how that fits into the story.
Right. Well, thank you. It's a great last question from a hero connecting two of my interests. So you won't have to twist my arm to think about how slavery plays into this story. And I would say, in the big picture in global history, slavery is an important, hugely important part of human demography and human connectivity. And that's also the story in the Roman Empire. And so the story of the city of Rome is inseparable from a constant influx of migrants. Rome is a death trap. I think it's a demographic sink, and it requires, we were talking about this with somebody earlier, we had great conversations with your very inspiring graduate students. There's 30, 40,000 people a year dying in the city of Rome, which is an environmental problem. How do you dispose of 30 or 40,000 corpses? But it's also a demographic problem. And one of the really exciting things that's happening now in the study of Roman demography is the use of stable isotope evidence to show that to confirm that the levels of in-migration to the city of Rome are huge, and it's not all voluntary. Involuntary migration of slaves is a huge part of the way that this demographic sink is continually replenished. And then the second way I think that slavery fits into this story is the connection across frontiers. And the example that I use with smallpox is the connection between Central and East Africa and the Roman Empire. And the slave trade is a part of that world. We know that spices, and I wouldn't begin to say anything compares with the spice trade. The consumer demand for that commodity is enormous in the 1st and 2nd century. But beyond that, gold, ivory, these supplementary commodities, slavery is right up there. And if you had to twist my arm, I would say that the Roman Empire is bringing in 5,000 to 10,000 people a year from Central and East Africa. We increasingly appreciate that it's not all across the waters along the Red Sea. It's also right across the Sahara, where the connective empire, the Garamantian Empire, is being literally pulled out of the ground. It's big. The trans-Saharan trade right across the Sahara is big in the 1st and 2nd centuries, and then around the Red Sea. And so the human connections built by the exchange of capital, by the exchange of commodities, including human beings, is a big part of the connectivity that binds people together and also binds together their germs and the way that pathogens can travel.
On that note of binding us together through our germs. Thank you.